Bismillahirrahmanirrahim. Disorders of urea cycle are dangerous because of the accumulation of ammonia in circulation. Ammonia is toxic to nervous system and its concentration in body must be controlled. Under normal conditions, free ammonia is rapidly fixed into alpha-ketoglutarate by glutamate dehydrogenase to form glutamate. Ammonia is further fixed by conversion of glutamate to glutamine. This reaction captures another nitrogen and prevents its escape into the circulation in the form of ammonia. Not only that, glutamate also serves to convert pyruvate generated in muscles by glucose metabolism into alanine by donating its amino group to pyruvate, thereby converting the pyruvate into alanine. Alanine then travels to liver as a non-toxic transport form of ammonia. Within the liver, as nitrogens are removed from their carriers through oxidative deamination, ammonia is released. In the liver, CPS1 fixes the ammonia to initiate the urea cycle by converting it into carbamoyl. When a urea cycle enzyme is defective, the cycle is interrupted, which leads to accumulation of urea cycle intermediates for the block. Because of block in urea cycle, glutamine levels increase in circulation, and because alpha-ketoglutarate is no longer regenerated by removal of nitrogen from glutamine, the alpha-ketoglutarate levels are too low to fix more ammonia. This leads to elevated ammonia in blood. Extent of elevation depends on the enzyme which is elevated. Glutamine osmotically draws water, leading to brain. Elevated ammonia interferes with energy generation by combining with alpha-ketoglutarate in the brain, which is an intermediate of the TCS cycle. Glutamate is an excitatory neurotransmitter. In the presence of excess ammonia, it is converted to glutamine, and the depletion of glutamate leads to lethargy and reduced nervous system activity. Cell death due to opening of the transition pores in the mitochondrial membranes of the astrocytes occurs in the presence of excess glutamine. Infants with defects in any of the first four enzymes usually appear normal at birth, but within 24 hours they progressively develop lethargy, hypothermia, and apnea. In contrast, arginase, which is the fifth enzyme of the urea cycle, its deficiency is not as severe as the deficiencies of the first four enzymes of the urea cycle. Let's have a look at enzyme deficiencies one by one. Infants with high ammonia, low arginine, and low citrulline levels must have a defect in urea cycle enzyme before the step that produces carbamoyl phosphate is not formed. At this point, one should keep in mind that carbamoyl phosphate is also formed by cytosolic CPS2 for pyrimidine synthesis, and orotate as an intermediate of the pyrimidine synthesis. So, in the absence of carbamoyl phosphate, there will be less than normal amount of orotate in the urine. Now, N-acetyl glutamate synthetase is another enzyme which synthesizes NAG, which is an essential activator of the CPS1, the rate limiting step in the urea cycle. NAG is synthesized from acetyl CoA and glutamate, and arginine is an activator for this reaction. So, in the absence of NAG, the rate limiting step of the urea cycle will be compromised. So the effects of its deficiency will be similar to the effects of the deficiency of the CPS1, that is, the formation of the carbamoyl phosphate will be compromised, and therefore the first step of the urea cycle is not going to occur, and the ammonia which was to be fixed by combination with carbon dioxide to form carbamoyl phosphate will not form the carbamoyl phosphate, so there will be hyperammonemia, and there will be low orotate in the urine, just as was the deficiency of the CPS1. The second enzyme of the urea cycle is the ornithine transcarbamoylase or OTC. In OTC deficiency, carbamoyl phosphate is produced because CPS1 is functional, but it cannot combine to form citrulline. It cannot combine with ornithine to form citrulline. More than normal amount of carbamoyl phosphate is diverted into the pyrimidine synthesis pathway, and this will result in the formation of the orotate going to appear in the urine. So orotate in urine and low citrulline are the hallmarks for the OTC deficiency, as this is in contrast to the CPS1 in which there were low orotate levels. The intermediates of the urea cycle before the block are elevated, and the intermediates which were to be formed after the block are low. So orotic acid urea differentiates between abnormality of CPS1 and OTC. In OTC, urine orotic acid is high. In CPS1 deficiency, it is low. The next enzyme is the argininosuccinate synthetase. Elevated citrulline and low levels of arginine suggest argininosuccinate synthetase or argininosuccinate lyase deficiencies. If citrulline levels are markedly elevated, it suggests that citrulline is not at all converted into argininosuccinate, and the defect is in argininosuccinate synthetase. If citrulline levels are moderately elevated, it suggests that some citrulline is converted to argininosuccinate, and defect is in argininosuccinate lyase. This shows that there is moderate elevation of citrulline, and thus this is a representation of the deficiency of the argininosuccinate lyase. And it is also pertinent to mention here that if argininosuccinate is synthesized, its levels will increase and it is going to be excreted in the urine, and as it is appearing in the urine, it is also leading to the excretion of the two nitrogens which were to be excreted in the form of urea. The fifth enzyme is the arginase. High levels of arginine and moderate hyperammonemia suggest that the defect is in arginase. So, moderate elevation of ammonia and low arginine are diagnostic of the arginase deficiency, and the signs and symptoms of this deficiency are not as severe as the sign symptoms of the deficiencies of the first four enzymes of the urea cycle, and the ammonia level in this enzyme deficiency is not as high as was present in the deficiency of the first four enzymes of the urea cycle. In nutshell, CPS1 deficiency is characterized by low urinary orotate, low blood citrulline, low blood arginine, and high blood ammonia. OTC deficiency presents with high urinary orotate, low blood citrulline and arginine, with hyperammonemia. Markedly elevated citrulline is hallmark of argininosuccinate synthetase deficiency, whereas argininosuccinate lyase presents with moderate elevation in citrulline, and in arginase deficiency there is moderate hyperammonemia.